about how we discovered DNA was a genetic material through the series of experiments. And now we want to talk about specifically how the DNA is replicated. So you guys should know that the DNA is a double helix, and it's held together by what? Two strands. So this is chapter 5 stuff. It's held together by hydrogen bonds, right? And there are different number of hydrogen bonds between the different nucleotides. So what does A pair with? T. G pairs with?
C.
How many hydrogen bonds between A's and T's? There's 2. How many between G's and C's? 3. So what happens here is that the 2 strands of DNA have to separate. So in the lab, we did this just using heat. But you can't do that in a living system. So how do we open the 2 strands? Right, so we change the shape. So imagine I don't know why. So imagine this is a double helix, right? It's twisted like a helix. So if I want to open this up, what do I have to do? Untwist it, which is, I can physically go in and do that. So there's an enzyme that recognizes a region that untwists the double helix at a specific site that we talked about during mitosis. And that site we talked about occurs in binary fission. Do you guys remember what that's called? It's the site of replication in bacteria in binary fission.
Cytoplasm?
It's called the origin of replication. So at specific sites of origin of replication, there's long stretches of A's and T's rather than G's and C's. Why would you want A's and T's at the site that you're going to open up instead of G's and C's? Yeah. So what do you think is weaker? 2 bonds or 3? 2. So there's an enzyme that's job is to untwist the DNA, untwist the helix. What would you name that enzyme if you discovered it? No.
Good guess.
Sure. What is it untwisting?
DNA.
Specifically?
A helix.
Okay, so
Helixase.
close. It's called helicase. So helicase unwinds the DNA at the origin of replication. Now remember that we talked about this when we had the 2 strands separated in lab. Then hopefully you guys talked about this, and if you don't have me for lab, that DNA is anti-parallel, 5 to 3 on one strand, 3 to 5 on the other. And what does the 5s and 3s represent? Do you remember? So it has to do with the chemical structure of the deoxyribose. So we number this, that we number the carbons. How do we number the carbons? Clockwise, right? So this is what number? 1. And what's on number 1? Do you remember? This is a nucleotide, so what's a nucleotide made out of? 3 parts to it. Have you guys learned this in the chemistry lab? It has a base, which is the A, G, C, and T. And so the base is found on the number 1, and then the number 2, what's on that? Okay, so I guess you guys just forgot everything for chapter 5, so I don't care. There's a hydrogen there on the number 3. Anybody know? No, it's a hydroxyl group. remember in RNA that's present, but it's deoxy, so that's removed. And then this is the fourth carbon, and this is the number 5 carbon, and what's attached to that? Anybody remember? It's the third part of the nucleotide, so nucleotides have a base,
Phosphate.
a sugar, and a, right, correct, a phosphate. So we have, and then this continues on to the next nucleotide with the next base. And so this would be the 3' end, and that would be the 5' end. So what's on the 5 here? That represents a carbon, so what's attached to it? It's right here. Right. What's attached to the 3? It's right here, yeah. So what do we call an OH group? Hydroxyl is on. So we have hydroxyl here and a phosphate group here. And we talked about the enzyme that copies DNA is called DNA polymerase, and it requires DNA polymerase requires double-stranded. Because it's an enzyme, right? And all enzymes have a specific shape. We talked about this in chapter 8, the lock and the key, right? Substrate, enzyme, they fit together. So the shape of DNA polymerase requires this to be double-stranded. So we've separated the strands, that's a single strand. Would this stay on here? If the shape is for double-stranded? No. So how do we fix that? How do we fix that in the lab? Okay. So we added a primer. So this sequence is then what would the primer sequence be?
C,
And so that's if this is a 5, that has to be a 3, so what's on this end? An OH, and if this is a 3, then this end must be a, and what's on this end? Excellent. Do you guys understand that? So the first thing on this bullet list is that the DNA helix must untwist, and that requires enzymes and proteins. What's the enzyme that opens up the DNA that untwists it? Helicase, right? It's named because of the helix. And then we have to put a primer down, which is a short complementary piece of DNA. In the lab, we use DNA, but inside your body, you use RNA. All right, and then obviously the new nucleotides that match these have to be directed into place. And then we want to make sure that they're accurate, that they've been put in accurately. And then we continue until we make a new strand. So the process of copying DNA is fast. It's about maximum speed, 500 nucleotides per second. And it makes one mistake every 1 billion nucleotides. So just to give you an idea, if that was your textbook, that would be one mistake per 10 stories of your textbook. Same font. So it's pretty good. Okay. So I'm going to redraw this. So helicase comes in and it opens the DNA. At specific sites, what are those sites called? Origin of replication, what are they composed of? A's and T's or G's and C's? Why?
Because they only have 2 hydrogen bonds.
Right. helicase, I would end up with a bubble, right? Looks kind of like a bubble, and then the two ends look like that. So what does that look like to you? Well, it's on a stick, so what would you call a U on a stick? It's a fork. So, okay, so we could call it replication Ys, but the technical name is a fork. So this right here is the replication fork, right? Because it looks like a Y. This is the bubble. Here's another replication fork. So for every origin replication, there's two forks and one bubble. I didn't make up the terms, but that's what it's called. So if when helicase opens up the DNA, what do the two strands want to do? They want to go back together, right? So how do you prevent two magnets from going back together? Stick something in between it. So what the cell does is it sticks something called single-stranded binding proteins that only bind to single-stranded DNA. And that keeps the two strands apart. And replication is bidirectional, so you can see that this fork goes this direction, right? DNA replicates this way, and it also replicates this way on both strands simultaneously. That expands the bubble out, which you can see here in step 2, and it continues to expand until they touch each other, and then we get two new DNA strands. So in prokaryotes, there's one origin of replication. And what's the shape for prokaryotic DNA? It's circular. Eukaryotic DNA, what's the shape of it? What do your chromosomes look like? They're straight lines, right? So eukaryotes are linear, prokaryotes are circular. And then this electron microscope graph shows you the bubbles, which are shown here in gray. So any questions about that? So let's recap. What's the first thing that has to happen for DNA replication to occur inside the cell? Okay, what untwists it? DNA helicase, where does it untwist it? Origins of replication. And what specific feature does the origin of replication have?
Two items.
It has A's and T's, right? So long stretches of A's and T's. All right, so helicase goes in, it untwists the helix. What's the next thing that has to happen? Right, and what's that something called?
A single
Stranded binding proteins. Right. So what do we create when it's open? What is that shape called? A bubble. In eukaryotes, there's multiple origins of replication because we have 1,000 times more DNA than bacteria. So think about it this way. You guys had 9 months to get from a single cell, a fertilized egg, to being born. If it took 1,000 times longer because you only had one origin of replication per chromosome, how much longer would you have to stay in your mother's womb?
forever.
Would you be happy about that? No, you probably wouldn't be born. So that's not possible because we have such massive amounts of DNA. So we have to have multiple origins of replication in order to be able to copy our DNA in a reasonable amount of time. So that's why it's showing you the multiple origins of replication. Prokaryotes only have one, right? So on the test, I might ask you, how do eukaryotes copy their DNA in the same amount of time as a prokaryote, even though it has 1,000 times more nucleotides? More than one origin of replication. Okay. So now that we've got the helix open, we've got the forks, we have to accommodate the DNA polymerase, which is the enzyme that copies the DNA. So remember I talked to my lab about Thermus aquaticus. That was the special enzyme that Carrie Mullis found in the hot springs at Yellowstone. So we need to lay down a primer, and let's say that this end is 3 prime, so what's on this end? It's a hydroxyl group. This one, if that's 3, what's this? What's on the 5? Excellent. This one, if that's a 3, what's this end? And what's on there? And what's on this end? Excellent. So I'm just going to make up a bunch of letters on here. I'll just say that this is A, G, C, T. Good enough. So if that's an A, what's on this string? And then perfect. Okay, so remember the polymerase can't fit on this. Why? Because it can't fit on single-stranded DNA. It can only fit on what? Double-stranded DNA. So what do we need to do to make that double-stranded? Well, yeah, but the polymerase can't start. So what do we need to put on there? Remember, we just talked about it. It's a primer. We had to put down a short piece of DNA, right? But I said in your cells, it's RNA. Okay, so let's say that we're going to lay down the primer right here. Your body's going to set down an RNA primer. So what end would this be? Well, okay, that's 3. What's this? 5? Let me make it a different color. Okay, so what primer would we order from the company? What do we want it to match this, so we have it? T. Is it a T?
No, U.
Yeah, because in our bodies it's made out of RNA, right? If we were going to order DNA, we would get a T, but this is a U because it's RNA, and then G, G, G. All right, we'll just stop there. So if that's 5 prime, what's on this end? Okay, so this is a phosphate, right? And this is a hydroxyl group. Now, the enzyme can fit on here because it's double-stranded. I want to stop it here, just so I don't have to redraw it. So let's see, this is the end, this is 3 prime. There's an O there. All right. I'm just going to make it scan the whole thing. All right, so which way is it going to copy this then? Or is it going to go both directions? Okay, so the DNA polymerase has to perform a reaction to add the next nucleotide. That we learned this in chapter 5. What's the reaction called? So if we want to build a polymer, what's the reaction we have to do? We want to make amino acids, we want to add glucose molecules to make carbohydrates, we want to add nucleotides to DNA, we want to make triglycerides. You guys did the chemistry lab, right? What did you have to do? Nobody remembers? You had to take water out, right? So what do we call that reaction? Dehydration reaction. Can you make water with a P? What's the formula for water? Tell me you guys at least remember that. H2O. Can you make water with a P? Can you make it with this? So then, can it go this direction? No, because it can't make water, and the dehydration reaction is required. So DNA can only be copied in this direction. Let's look at the other strand, right? So we would have to do the same thing. Your body would have to do the same thing. It would lay down a primer. So this primer, if that's a 3, what would this end be? 5. I'm just going to start here. So that T would pair with A, and then G, A, U, C, C. I'll just end it there, and let's say that that's the 3' right? So again, the polymerase requires it to be double-stranded. It can't fit on it otherwise. So let's say that there's another polymerase that comes along and sits on here. It doesn't know any different. As long as the requirement is there, it'll bind. It doesn't matter if this is a human or a mouse or a monkey or a bacteria, it's still going to bind like this. So as long as this requirement is there, the polymerase will bind. And then, so what's on this 5' end? What's on that end?
Phosphate.
Right? And what's on this end?
Hydroxyl.
Hydroxyl group. So which direction is the DNA going to be copied here?
Hydroxylate.
So it goes this way, right? But I told you guys that DNA copies on both strands in both directions simultaneously. So did I lie? Maybe. So it cannot copy that direction. It's against the laws of chemistry for that to occur. So, but there's a way it gets around it. Any thoughts about that? All right, I'm going to let you guys contemplate that while we go back to this slide and talk about where the energy comes from to move this thing along so that it can copy DNA. So the nucleotides themselves are their nucleotide triphosphates. Where have you seen that before? What's our energy molecule? ATP. What's the A stand for? Adenosine, right? So what is that? It's a RNA, which is a nucleotide. How many phosphates on it? 3. So because this is DNA, so a nucleotide triphosphate would be NTP, right? That could be ATP or what's the other letters? GTP, I'll help you out, and or CTP or no T's and then DNTP, so that stands for DNA, would be D ATP, D GTP, D CTP, and D TTP. So this has energy in it, right? We already know that. We spent a whole chapter learning that. Do you think there's energy in that? Yeah. What's the difference between this one and this one? Yeah, it's this. So yeah, there's energy in that, and the energy to move the polymerase along comes from the nucleotides themselves. So this is just showing you that in order to make the reaction occur, we need an OH group, a hydroxyl group. I think you guys understand that. 2 of the phosphates get cleaved off, so the phosphate gets put together like that. This is showing that a T is pairing to an A. So any questions about that? All right, let's recap. So what has to happen to initiate DNA replication? What does that unwind it? Helicase. Where does helicase open the helicase? Origin of replication, what's it made out of? Why? 2 hydrogen bonds. Okay, so we have it open, what's next? It has to stay apart, so what do we use? Single-stranded binding proteins, excellent. So now we've got it apart, what's the next thing we have to do?
The primer.
We have to put down the primer, right? What does jobs inside our cells?
What is it?
What does jobs inside our cells? Enzymes. So what do enzymes end in? ACE. So what would you think puts down the primer? An enzyme, what would you call that enzyme that put down the primer? Primase. So that's the name of it. So primase comes along and lays down a primer. What's the primer made out of? Right, so what do we call those together? Collective RNA. Good. Now, the primer provides 2 things for the DNA polymerase. It provides the correct shape. Polymerase requires double strands. And it also provides that OH group where it can add to. You guys see that? So without that primer, the polymerase couldn't copy DNA because it wouldn't have the right shape and it wouldn't have a place to do dehydration. So that's what the primer does. Then the next thing happens is what? What replicates it? What does jobs in the cell? So what's the enzyme that replicates DNA? It's right on the very top of this slide. No. Do you read the same slide? DNA polymerase. Yeah, that's the enzyme. All right. So remember that we talked about this. So DNA is anti-parallel. That means that one strand is 3 to 5. The other one is 5 to 3. And that presents a problem because you can't replicate DNA only in one direction, which I think that you guys get. So because there's only one free 3 OH group on a primer. Does anyone have any questions about this and how DNA can only replicate in one direction? All right. So we talked about the primer is laid down by the enzyme primase. What I didn't tell you is that the enzyme that specifically copies, this one I've drawn on the board in red, is called DNA polymerase 3. So there's a 1 and there's a 2 and there's a 3. Why do you think it's called 3? So remember, it's the same reason that the photosystems are numbered the way they are.
There's a third one to be found.
It was the third one to be found. That's correct. So even though it's the one that does the most work, it's still named 3 because that was the order it was discovered in. So on the test, I'm going to ask you to distinguish between 2 different polymerases. One is a polymerase 3 that does the main copying, and then there's another one that we have RNA, right, in our DNA now. So do we want to keep RNA in our DNA? Why not? What's wrong with RNA? You guys are RNA haters?
It just does it only does the replication. It's the computer.
Well, it's not that's true, but that main problem is that the RNA is different in DNA. We talked about this in chapter 5. I'm sorry.
It has the U.
It does have a U, which is a close, remember we talked about this in chapter 5, the difference between the U and the T is that methyl group. But there's something more important. What about, how long does RNA last versus DNA? So I'll give you a hint. They just pulled DNA out of a Tyrannosaurus rex femur bone that was 125 million years old. But you guys make RNA when you go outside and it's cold to shiver. So how long does that last? Forever? 125 million years? Minutes, right? So remember, RNA is inherently unstable. Would you want something that's unstable in your DNA holding it together? No, that would be bad. And why the cell uses RNA instead of DNA, right? Because this seems inefficient, right? Put RNA in and then take it back out and put in DNA. But nobody really knows. So you guys want a Nobel Prize? See if you can figure that one out. But we have to get this out, and so the enzyme that removes this is called DNA polymerase I, and it replaces it with DNA. So it's showing you here. Primer is put down by what enzyme? Primase. Now the conditions are met. What's the direction that DNA is copied from the primer? 5 to the 3, right? And so it continues copying. This blue is showing you, this light blue is the new DNA. This is the dark blue is the old strand. Here's the enzyme DNA polymerase. What's the name of this polymerase? 3. And now there's another polymerase that comes in and replaces the RNA in the primer with DNA, and that is polymerase 1. So now we have a new complete strand of DNA. We get it?
How do you tell the difference between 3 and 1?
So 3 has one job and 1 has another job. It's like, how do you tell the difference between someone that puts on tires on the car and someone that puts in the engine? That's just what they do. And they have different shapes, so they have different jobs. Does that make sense?
Yeah.
Okay. All right, so back to our problem. I told you guys that we copied the DNA in both directions on both strands simultaneously, but that seems literally impossible by looking at this, doesn't it? And how it does it is that there's a trick that we can lay down a primer and go in this direction, right? So if it's moving this way, what we have to do is, where's orange? Is that this one, it can continue to be copied. So this strand is called continuous, or it's also called the leading. That's a slash. Strand. And this one can be copied all the way, right? So this is continuous and leading as well. Does everyone see that? But this one can't physically go this way. So what it does is, as this one's approaching, this one lays down another primer up here. And then if this is 3, what's this end of this primer? 5, and this one is 3. So it can synthesize a new strand like that. So what it does is it lays down a primer and copies it backwards, and lays down a primer and copies it backwards, while this one is just barreling along. Does that make sense? So it makes it in these little pieces called Okazaki fragments. Because who do you think discovered them? No, his name is Fragment. Scientist joke. So the problem is that no polymerase can link these two pieces together. It doesn't have the ability to do that. So there's another enzyme that has to come in here and seal each one of these gaps between the primer and the new piece of DNA. And that enzyme that seals all these is called ligase. So Okazaki was able to discover these pieces because the bacteria that he was looking at didn't make this enzyme correctly. So what would that leave in the DNA then? These little short pieces, right? Because they were never stitched together. So that's how he discovered these fragments, and that's how he discovered how the other strand gets copied. So if this one's continuous and leading, then what would you call this one? That has to be made in little pieces. So this one is called discontinuous or the lagging strand. So on this fork, the top strand is the leading or continuous strand, and the bottom strand is the lagging or discontinuous strand. But over here, the bottom strand is the leading or continuous strand, and the top strand is the lagging strand. Do you guys see that? Okay. All right, so this slide is just going over the steps. So let's go over them really quick again. What has to happen in order to copy DNA? Helicase comes in, recognizes the origin of replication. What's the origin of replication that makes it unique? A's and T's. Why? Two hydrogen bonds. So it opens it up. Now the strands have to be held apart. So what does that? Single-stranded binding proteins. Then what's the next step? Primer gets laid down. What enzyme does that? Primase. So once the primer's laid down, it fulfills two requirements of the polymerase, which is one, it's double-stranded. Two, it has a OH group, right? And then what happens next? DNA polymerase binds to that and starts copying DNA. What polymerase is that? 3, right. So what's the primer made out of? It's RNA. We don't want RNA primers in our DNA, so what do we have to do? Take it out and replace it with DNA. What enzyme does that? DNA polymerase 1. So all these questions I'm asking you, you can expect to be on the test. All right, so this is showing you what's the priming for the leading strand, primase. What's the priming for the lagging strand, primase. Elongation is done by DNA polymerase. What's the name of that polymerase specifically? 3. Replacement of the primer, RNA primer by DNA is done by DNA polymerase. What specifically? 1. Excellent. And then what joins the fragments together? Ligase. Good. All right. So let me show you guys an animation of this in action.
Its strands are separated by the enzyme helicase. DNA replicates. Its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from reannealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase 3. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then DNA polymerase 3 lays down new DNA. The process repeats again and again.
So what's this thing called right here? Bubble, and this right here where all that junk is? Fork. Good.
It's straight.
All right, so that's how it works. Any questions about DNA replication?
You said the primer
provides double-stranded DNA because the polymerase can only fit on double-stranded DNA. All right. So there's a problem that occurs in our DNA, and that is that every time we replicate a linear chromosome, it gets shorter. And at some point, it gets so short that it starts cutting into genes that we need to control the cell cycle, and it causes the cells to undergo apoptosis. You guys know what that is, right? Cell suicide or cell death. So cells can only divide a certain number of times, and then they undergo cell death because of this shortening of the chromosomes. And this is why it physically happens. So here's the parental strand of DNA. Here's the end of a chromosome. The end, do you guys remember what end means? What's the Greek word for end? We talked about it in mitosis and meiosis. Telo. And then what's the part?
Mer.
Right, so telomer is what we call an end of a chromosome. Yeah. So this is the telomere right here. And so here's the leading strand. The primer is laid down. The leading strand is copied continuously. Here's the lagging strand, so it's copied in parts. Here's a primer, here's a primer, here's a primer. So you can see the lagging strand can go all the way to the very end of this new chromosome, this newly copied chromosome. By the way, where does DNA get copied? What phase of the cell cycle?
In the S.
S, yeah. So all of this occurs during S phase. And so now we have to remove the primer, right? So the enzyme comes in, removes the primer, but what's the problem here? It can't add nucleotides to this end because it's a phosphate. So the leading strand, every time your chromosomes are copied, every time you make a new cell, gets shorter and shorter and shorter and shorter, right? So think about it this way. Let's say that you have a 20-foot driveway and some joker comes up every night and cuts off a foot of your driveway. And there's nothing you can do about it, right? Nothing. What would you do to prevent your driveway from getting shorter? You can't get rid of the joker. It's going to happen. There's nothing you can do about it. You can't build up a wall. You don't want your driveway to get shorter. What would you do? You just make it longer, right? What if I added 20 feet on my driveway? I wanted to keep my original 20 feet, right? So the joker comes and chops up the junk that I don't even care about. So there's an enzyme called telomerase that does that. What it does is it just adds junk on the end of the chromosome. So let's say that here's the end of our chromosome. Right here, here's the telomere. And we know on the leading strand that every time that gets copied, it's going to go shorter, right? So nothing we can do about it. It's just a physical property, chemical property. So this enzyme telomerase, what it does is, so let's say that this is an important gene. Once we hit this gene, we're going to undergo apoptosis, right? So let's say 1 replication, 2, 3, 4, 5, 6. Now we're getting into the gene, right? So what telomerase does is it just adds a bunch of junk on the end, right? So now, how many replications do we get before we end up in that gene? A lot more, right? Does it matter what this is? It could just be random A's and G's and C's or whatever. So telomerase's job is to extend the end of the chromosome to make it so that it doesn't matter that it gets cut. And guess what this does to cells if you have really long ends of the chromosome? You think it makes something live longer? What's one of the properties that cancer has? It's immortal, right? Well, all cancers have telomerase active. It's all turned on in all cancer cells. And that's what makes them immortal, right? So if I took any cell out of your body, put it in a petri dish, it would divide like 50 times, and then it would die because of apoptosis, because of this. But if I took a cancer cell and let it divide all day and night, it would never die. Why? because it has these massively extended chromosomes. Why do we have telomerase? Well, think about it this way. You guys came from a single cell, right? So would you make it out of your mother's womb after 50 divisions? So when you're an embryo, you have to have telomerase turned on. There's no way you can make it after 50 divisions. But once you're born, it's turned off. Why is that? Not all cells. Some stem cells have it on, but most cells have it off. Why is that? What does having telomerase on lead to? Cancer, right? So there's a fine line here between immortality and developing cancer. So even if you could live forever, right? Even if we talked about these Raelians, right? And we could clone sheep, but when we do the clone, what does that clone already automatically start with? Shorter pieces of DNA, right? So the clones of organisms don't live as long as the parents because of this telomerase. So cancer biologists are really interested in telomerase because if you could turn it off in cancer cells, what would happen? They die. They die natural apoptosis death because their chromosomes would get short. All right. So any questions about telomerase or telomeres? All right, so the last thing we're going to talk about is how DNA only makes one mistake per billion nucleotides. And it's really not that good. You know, think about you if you're typing a book. You're going to make a few mistakes, right? But let's say that you had a bunch of proofreaders that read your manuscript before you were going to publish it. There would be less errors, right? The more people that read it, the less errors there are. And so we have a battery of enzymes that their job is to proofread the DNA once it's copied to make sure everything's okay. Just like someone proofreading your novel or whatever. So if you didn't have these proofreaders, what would you think would happen?
Be a lot of mistakes.
Right, you'd have a lot more mistakes than you would normally have. And the way that these proofreaders work is they detect subtle changes in the shape of DNA. So let's say if I had a growth on my arm, how would I know that? I could rub my hand across it, right? And I would feel it because my fingers would move up. And so the same thing, DNA has a specific shape, so these enzymes go down the DNA, and if they feel something unusual, like a bulge, then they go, uh-oh, there's a mistake, and I need to correct that. Does that make sense? So those enzymes are proofreading enzymes. And we talked a little bit about what happens when you lay out in the sun too long. You guys remember this? So if you have two T's, I didn't draw any T's. I didn't draw two T's. But if there are two T's next to each other, they would bind, they would covalently link to one another. And so if you have, so imagine this is the backbone of DNA. You have two T's linked together. What would it cause there? It would cause a bulge, right? And that could be detected by these enzymes that are scanning the DNA. And that's shown here. So this is called a thymine dimer, which is caused by ultraviolet light. You guys go out in the sun, it happens. There's nothing you can do about it. Any ultraviolet light that hits your DNA and there's two T's in it, this happens. So how do you get rid of it? Why aren't we all dying because our DNA is all screwed up? Because we have enzymes that can repair it, right? So the enzymes come in and they make cuts on either side of the bulge, and then they remove all of those nucleotides. So the enzyme that does that is called a nuclease. And there's two kinds of nucleases. There's an endonuclease and there's an exonuclease. So what would you call one that made the cuts on the inside? Endo. And if it was cutting from the end of the chromosome, it would be an exonuclease. So that's the difference. So this particular nuclease is an endonuclease. Its job is to cut the DNA and remove it. Because it's a covalent bond, is that reversible? No, so you just got to dump it and start over. And then the enzyme that lays in the new nucleotides is the same one that replaces the RNA primer. So what was that called? No, the one that replaces the primer, not puts it there. DNA polymerase,
3.
no,
2, 1.
1, that's right. So DNA polymerase 1 replaces that. And then, remember there's a gap, so what enzyme can seal that gap? Ligase, right? So that's the enzyme. So make sure you guys know these enzymes and what their role is, because I'm going to ask you that on the final exam. And in fact, it's interesting because there's a disease called xeroderma pigmentosum, and that those people don't make the nucleases to recognize and repair thymine dimers. So what do you think they can't do? They can't repair thymine dimers. It's also known as vampire's disease. What do you think that is? They can't go out in the sun because it will kill their cells that they can't. So any amount of UV causes them to have an instant sunburn. It can't be repaired. And that's caused by one mutation in one single gene.
It's not another ingredient.
No. No. I can pull it up and show you what it looks like real quick.
Tuesday, do we have class or no?
Next week?
Yeah.
No, it's finals week. So you guys will have, if you have other classes, you may have a final exam then.
Right.
The final exam schedule is posted on the calendar link on
So
their
this is pretty much like a review class day.
You have
Yeah, we
class
have class
Thursday, yeah.
Oh yeah, I'm sorry, I'm thinking
Yeah.
today is Thursday.
No, it's not
Other
Thursday.
than that,
I
Yeah.
have a question on the telomerase. You said it's active in the womb and in the stem cells,
Right.
so it's not active in our usual and regular old cells?
So no, it
No,
isn't.
so it doesn't elongate the
No.
DNA on the...
No. So this is what this looks like
Turn
in
this
people.
back on.
Um, So it causes them to have higher incidence of skin cancer, right? And you know, peely, blotchy skin. This isn't holes in their face, it's just blocked out for some weird reason. And then you can read about it right here.
So those blotches are in sunburn.
Right. Okay. So that's the end of chapter 16. Do you guys have any questions before we start into 17? Okay. So this will be the last chapter that you're responsible for the final exam. And so this chapter has to do with how we go from DNA to protein on a molecular level. So you guys know that inside the nucleus we have alleles, and those alleles are expressed and converted into what? What are our genes turned into?
protein.
Right, and so what's the intermediary? Because remember, we only have one copy of our DNA, so we don't want to just throw it out in the cytoplasm. So what do we make instead?
And the mRNA.
Right, we make mRNA. So in this chapter, we're going to talk about how DNA is turned into RNA, and specifically this is mRNA or messenger RNA. And then that RNA is turned into proteins. So you guys know a little bit about what makes mRNA into protein. What are those things called?
Amino acid.
Chapter 6. No. So what's the machinery that turns mRNA into protein?
Ribosomes.
Ribosomes. Excellent. So how many letters in DNA?
4.
How many letters in RNA?
4.
So that's basically a similar language, right? So when a court reporter is taking down notes from a courtroom, they're doing it in a similar language. So what do we call that?
Transcription.
Right, so this is transcribing or transcription. And then how many letters in RNA? And how many letters in proteins?
20.
20. So that's a completely different language, right? So what do we call it when we convert one language into another?
Translation.
So going from DNA to mRNA is called transcription. Going from RNA to protein is called translation. All right, so we've talked about this before. Do you guys remember what this whole process that we talked about is called? It's the central dogma of molecular biology. And so the question is, all of our cells have the same DNA. I think that we established this early on, right? So what makes a heart cell different from a lung cell, different from a brain cell? What's it?
DNA.
Right. So the DNA's turned on or off differently in different cells, right? It responds to the environment different ways. And so the question becomes, what controls whether the DNA is made into RNA or not? And in order to understand that, we have to understand what the regions are of DNA that are turned into RNA. So that's what we call the alleles, which sometimes are called genes, right? Mistakenly called genes. So that's what makes up our genes. And so I think this is just a reminder, but you guys know that transcription takes DNA, makes it into mRNA, and then translation makes that into protein. And then prokaryotes, that occurs all in the same space because prokaryotes don't have a nucleus, right? But in us, transcription occurs where? In the nucleus, and then translation occurs where the ribosome's found? In the cytoplasm, right? So there's two different spaces that this occurs. In the nucleus, transcription occurs, and then in the cytoplasm, translation occurs in eukaryotes. It's all together in prokaryotes. All right, so I'm just going to revert back to the terminology that you guys are probably used to, and we're going to call an allele a gene just for this chapter. All right, so what are the characteristics of a gene? So let's say that we have chromosomes, right? And let's say that this one is for, this gene is for eye color, and this particular one is for blue eyes, so it doesn't make anything, it's defective. This one is for brown eyes, so it makes a brown colored protein, right? A protein that reflects light at the brown wavelength. Does that make sense? So we're not going to look at this one because it's broken or defective. This is the thing. This is the default. So let's say that this is the brown gene, even though it's an allele. So here's our gene. And so this gene actually has three parts to it. One part is called the promoter. And the promoter is like an on and off switch. In fact, it's more like a dimmer switch. So you can have it on a little bit, or all the way up, right? Or halfway, or just a tiny bit, or off. And so in that way, we can make lots of RNA, or just a little bit, or whatever we want. And then there's a region called the coding region. And that's the region that the ribosomes are going to look at, and they're going to turn that into a protein. And then, so there's an enzyme that does this called RNA polymerase. So the one that copies DNA is called DNA polymerase. RNA polymerase comes on here and it looks at the letters. So let's say that this is an A and a C and a G and a T and an A and a G and a C and a T and so on, like so. And in genes, again, they're going to be they'll have 3' and 5' ends. This is only one strand of the DNA. Okay, what's on the 3' end? Good, and the 5? Excellent. So when the RNA polymerase sits on here, it's going to read this, and what's it going to put when it sees an A? Remember, if this is a 3, what's this end going to be for the RNA? That was a 3, what's this new RNA end going to be? 5, right? So this would be 5', and it sees an A, and what's it going to put in there? U? And then this would be the 3'. Okay, good. So this thing's going to keep reading, right? So what tells it to stop? What tells it where the end of the gene is? And that's a good question, and I'm glad you asked. So it's an area called a terminator. Okay, not the governator. But it's a physical piece of DNA that looks like a stem in a loop. So you can think of the RNA polymerase as a train rolling down this track. How do you get a train to derail? You just put some big thing on the rail, right? And it's going to jump the track and fall off. So this actually has a physical stem and a loop to it that causes the polymerase to hit that and fall off. So the promoter is where the RNA polymerase binds. The coding region is what is turned into protein. Kind of. And then the terminator is what stops transcription. So all 3 of these, promoter, coding region, and terminator, constitute what we would call an allele or a gene. And that gene would be for a protein that makes your hair brown. Does that make sense? So, you want different genes to be expressed differently, right? Let's say that you guys know that you make genes that break down glucose, right? So, and you also make genes that code for hemoglobin that make the protein that carry oxygen for your blood. Would you want those genes to be on all the time, you think? I would, you know. So those genes aren't regulated. They're what we call housekeeping genes. They're always on. There are certain genes, so this is the beta subunit of hemoglobin. And so there's a little code in here that says, turn this on a lot, right, in the promoter. This one is SV40. This is a simian virus. So if you're a virus, would you want your genes to be turned on all the time? Heck yeah. You want to get in there and get out before you get detected and killed, right? So you want your genes to be screaming whenever that you infect a cell. So this has a different promoter thing so that it's on really hard. What's a kinase?
It's a phosphate. It
It adds a phosphate, right? So do you need kinases all the time? Do you need them active all the time?
Yeah.
Probably not all the time, right? Maybe a little more than normal, but not all the time. And then this is for histones. This is a subunit H2B. Do you need histones all the time? When would you need to make new histones?
It's the whole DNA together.
Right, so when would you need to make new histones?
only an S phase. when
Right,
you're
so
looking at
only if you're replicating your cell would you need to turn this on. So do you guys get the point that different genes need to be turned on or off at different levels, depending on what their job is? And so they have different codes in the promoter that let them know, hey, I should be at this level of dimmer, or this level of dimmer, or this level of dimmer, or off or on, all the way. Does that make sense? So these little codes, remember the DNA, all it is it's letters, right? It gets an instruction set. So the letters tell the polymerase how often it should be expressed or how it should read this. So some of those codes are what we call the TATA box, which is, because its letters are TATA, right? And so that's shown here in the pink. And you notice that everything here except viruses have a TATA box. So that's the main thing in eukaryotes, is that that tells the RNA polymerase where to bind and line up. And then there's other ones. One's called the CAT box. Why do you think it's called that? CAAT. There's a GC box that has lots of G's and C's. There's octamers. So there's different things that are found in different areas. The one we're going to focus on is this TATA box, though, because that's the most important one in eukaryotes. And so we'll pick up the 3 steps of transcription on Thursday. I don't think we'll have time for a review, but what I told my other class is that I'll take an overview from spring for the final, and I'll put it online so you guys can watch it on your own. Alright, so if you guys are lost, maybe you might want to just browse chapter 17 for Thursday, and that'll be our last regular class meeting. I'm not sure if we'll get through all of chapter 17, but I'll try. And then your final exam will be a week from Thursday in this room at, I think, the same time, or maybe 8:35 or something ridiculous. So I'll be in here at 8:30. If you want to start your final, you can. It'll be the same format, 3 chapters, chapter 14, 16, and 17. 50 questions. You have 2 hours, but it shouldn't take you that long, right? It'll probably take you less than an hour. All right, so I'll see you guys on Thursday.
And the Tuesday night class people, when you do the desert botanical garden as extra credit?
No, because it's not fair to the other students, because they can't really get that as an extra credit, because it's a required thing.
Oh, I thought the mitosis.
I did mine.
Because I thought you said in the beginning.
Yeah, you said you don't need a, we could, but then,
You guys, so I told you guys that you could do the other one, the one where you walked around and identified.
Oh yeah,
the-
that's the one I was thinking.
Yeah, so if you turn the botanical garden in,
can't get extra
I'll
credit
give
for
you the
that.
5 points instead of the other one.
All right. Gotcha.
But make sure you turn it in under the one that says Unit 14. Because there's 2. One
There's
is
a 5
Sonoran
for
Desert
areas.
Plants, right? And then one is Desert Botanical Garden. So students get those mixed up all the time. But one is from your lab book.
So just copy it under the,
Yeah, so just take pictures of it or whatever and submit it.
or I could just redo it.
Wait, so which is the one we can do? The-
So you should, for extra credit, you should do the one out of the lab manual, which
The
is
plant
Unit
one,
14,
where we had to.
where you identify
figure
the
out
plants.
all
So
the
it's
plants
just
that
one
have them.
page, it's page 5.
Oh, okay, so not the botanical garden.
Right, so because that's worth 25 points, and my students didn't have a lab, but you guys went in and did that, a lab. You did the walking around and identifying the plants already, right?
Well, we just had plants that
It's the same
we
thing
have
though,
it.
they're the same plants.
Oh, the
Yeah.
mustard.
It's the same.
So,
just
so
the
you
same
guys,
sheet that
you
we-
guys would get 25 points for that lab, where the other class wouldn't be able to get that 25
Is-
points because,
But,
Because we have what, links on our list of assignments on there, so like, is that, if that gets wiped out, does our grade look better?
is it just not
Because on
factored in
Canvas,
for
there's
us?
like, we don't have any points under those assignments. Because we're in the other.
right, you guys will get points for being in class that day. Because my students didn't have
Oh, okay.
a class that day. They were supposed to go to the botanical gardens and do that instead.
So are you judging it off attendance, or are you judging it off our grade there? Because he gives us like lab reports and
And quizzes and a
and.
paper.
So, yeah, I mean, this is the problem of having separate instructors for lab and lecture. And it's inherently bad, and I've sort of talked to them about that, but they'll only let adjunct teach a certain number of hours, because, then they would have to be paid
More.
health and stuff, and they don't want to do that, and they will only let me teach a certain amount of hours, so it kind of gets screwed up.
Yeah.
And I realize that. So the best I can say is that I will make adjustments to try to make everything fair for both labs, which is what I'm trying to do with this extra credit thing too.
Okay.
Because it's not fair that you guys would get to double dip, go to class and then turn in something where my students wouldn't be able to get those extra 25 points.
But they just have to show up for class though, right?
Well, yeah. But I mean, that's because I do things differently. So at the end, I will look at the grades he submits to me, and then I will adjust them to be more in line with my lab grades. So that's all I can really tell you, and you're just gonna have to trust me on this, because I can't, make him do stuff, because there's a thing called academic freedom that protects the way people teach. But ultimately, I'm the one that submits. Is the stuff recording? Yeah, I'm gonna stop this.